And I'm going to talk about analyzing cortical features via their distribution functions. So this is joint work with Ian Bowman, Alfred Doga, and Jack Van Horn at London UCLA. So why are cortical features such as volume, thickness, area important? So one reason is you can understand the global structural patterns for a given subject or even across population by understanding how these individual features vary across the brain. The second reason is there's specific cortical features implicated in disease. For example, here, so here on the bottom left corner, you have a result showing a decrease in the cortical volume by Shire and et al. So they studied children with congenital heart defect, and they found out that the patients had a decreased volume, cortical volume compared to normal controls. In this case, Lehman and Crutch studied thickness, and they found thickness deficits for persons afflicted with Alzheimer's. In this case, Lurch and Evans, they also found thickness reduction in case of Alzheimer patients with respect to normal controls. And in this result, Goldman and Fischel, they analyzed cortical thickness changes for patients with schizophrenia and found significant deficits in thickness over across the brain. So they also conducted heritability studies based on the same population. That's why cortical features are really important, so that they can tell us something more as to how the disease is either getting propagated or affecting the different parts of the brain. Thirdly, there is a lot of emerging interest in inferring statistical brain connectivity networks. So we have a lot of work done on actual work done on extracting the actual structural connectivity in the brain, and that involves tractography, diffusion weighted imaging, and so on. There's another new emerging field that instead of looking at tractography and structure derived from tractography, wants to infer structural interdependence between different features in the brain. And so you have this work by Hay and Evans, even Hagman and Spons, who've actually analyzed features such as thickness and volumes, and try to infer a statistical network that shows you how volumes or thicknesses vary and depend from one region to the other in a subject and across a population. We have also done a similar study where we've actually used correlation and partial correlation on thickness measures for a brain, and a simple distance matrix based on a correlation of thickness without doing any tractography can actually discover many interesting connections in the brain. So this is purely based on MRI images and no information about tractography at all. So these are a few cortical features: thickness, sulcal depth, and curvature overlaid on the brain with a color-coded map. And so thickness and sulcal depth is strictly positive, whereas curvature can also assume negative values. So what we are interested in doing is dealing with cortical representation for the brain, parcellating it into different regions based on either functional atlases or any other atlas that you might have. Measure different features such as the cortical area, thickness, volumes, curvatures, functions of curvatures such as shape index, curvedness, etc. for a region, and analyze the variation of those features by using distributions. So in all of the work that I mentioned earlier, people have used average values of these features: average thickness, average volume, average area, surface area. What we want to do, however, is to analyze the full distribution of that feature along the region. So you have a cortical surface parcellated into different regions, and you can denote a spatial parcellation by a map from R2 to R3. It's a simple geometric representation. And so the cortical feature is the scalar function actually projected on this 2D domain. And it can be thickness, volume, whatever you want. So in this case, I'm showing histograms of thickness for each, for a few regions of this brain. So we use relative frequencies of this variation of this feature and convert the 2D variation to univariate distribution, which will be the function of choice for us. And so this variation is the 2D variation. We converted it to a single univariate representation. And what we want to do is to have a non-parametric density estimate based on this distribution. And so we use a method recently proposed by Botev, who use a Gaussian kernel density estimator for a given distribution. And so they use the fact, and they were not the first ones to prove it, that this estimator here is actually the unique solution of the heat equation, which is shown here. And so what they do is they have a diffusion process, and they iteratively solve this PDE to arrive at your density estimate. And this is the density estimate that we will use or construct for each of the cortical parcellations, features of the cortical parcellations. So these are a few cortical feature patterns that you can see for different features: thickness, volume, area, and curvature. And so for a given subject, for a given brain, the cortical feature pattern is actually a set of these density functions, PIs, for n regions. And so for blowing up the thickness and the area patterns in detail, you can see that not all of the patterns for thickness are completely unimodal. They don't even appear Gaussian. In fact, thickness does not really look very much like the area. If you actually viewed area and volume, they look pretty much similar. But thickness is very different altogether from both area and volume. And so we think it's giving us some more information which is not directly gleaned from area and volume. So given this cortical pattern or this feature vector, we want to have a choice of statistical measure for comparing distributions. And so there are many choices here. One is the Kullback-Leibler, which is the difference in the entropy between the two distributions, whose densities are Pk and Pm. Then there's the Bhattacharya distance and the Hellinger distance, which are actually using the square root of the density functions. And then there are Earth movers distance, which is going to compute a flow between one distribution to the other. And a simple Euclidean distance, where if you consider Pk and Pm to be simple functions in a Hilbert space, and you simply take the Euclidean inner product, which is the distance. In this work, we use Jensen-Shannon divergence, which is a symmetrized version of the Kullback-Leibler divergence, essentially computing the difference in the relative entropy between one of the distributions and the average of the two distributions that are in hand. And it has got some nice properties. It's greater than or equal to 0. It's symmetric, and it's equal to 0 if and only if Pk equal to Pm. And so the cortical feature distance is finally given by a weighted sum of all the Jensen-Shannon divergences for all the regions for both of the hemispheres. So we know that this is not a true metric. It doesn't satisfy triangle inequality. But the square root of this form is actually a metric. But however, in this work, we've used this form as presented. So finally, I'll discuss a few applications and results based on the approach of using distribution functions for features. And here's our data. We have a big data set of multiple subjects collected at different sites. So you have the ADNI data set, and we chose 117 subjects out of that data set. Then you have the Australian ADNI, AIBL, 236 subjects. You have the Consortium for Neuropsychiatric Phenomics, which was collected at UCLA, and the ICBM, which consists of just entirely normal control subjects. So we look at the ADNI data set first. So what we did here was, for each of the brains, we had a cortical parcellation and computed those features, and computed a distance matrix based on the Jensen-Shannon divergence. And this is the actual distance matrix for the 117 subjects. And then we did a hierarchical clustering based on average linkage and found a dendrogram. So let's view this dendrogram in more detail. So we just threshold this dendrogram. So we didn't do we didn't use any other classification methods here. So we just threshold this dendrogram into two groups, the Alzheimer's and normal controls, and we leave the MCIs as it is for now. So we found that simply based on the thickness and without doing any pairwise registration, you could actually classify 80% of the Alzheimer's. So they actually coherently group together with the Alzheimer's group. Whereas for the normal controls, you have a 90% success rate of normal controls getting clustered with their group. Whereas the MCIs are scattered throughout, which is not entirely unexpected, since to really classify mild cognitively impaired subjects, you need a longitudinal element to the study, and which we haven't done here. We compared the cortical feature matching with Euclidean distance, a simple inner product between the two density functions. And this time it did not fare so well. So for the Alzheimer's, it probably improved. You could classify 85% of the Alzheimer's, they stayed within their group. But for the normal controls, around 40% of the normal controls were actually misclassified. And I think this is because of the choice of the distance between distributions that plays an important role in how these patterns actually get matched eventually. So you could also use this for visualization, and these are the same 118 ADNI subjects, 39 Alzheimer's, 38 MCIs, and 40 normal controls. So what we did was we projected the pairwise distance matrix into a 3D space using MDS and simply color-coded them according to their labels. And so you could see a nice separation of the normal controls and the ADs, and MCIs are all again scattered throughout the data. Lastly, what we did was we pooled all of the data together, and this data consists of multi-site trials, different scanning parameters, different scanning protocols, different collection protocols, different imaging protocols, different geographical locations, of course. So just based on the distance matrix, again, we projected it in the 2D space this time using MDS, and you could see that the sites have now fallen out very easily out of this analysis. So you see all the ADNIs being clustered here, all the AIBs kind of clustered here, and the CNPs and the ICBMs. ICBMs are all completely normal control fellows. So the important lesson here is when one integrates multi-site studies, one of course knows to correct for the multiple sites, but it's not so straightforward as to how to do that exactly yet. And so the object of this study is, of course, not to use all of these data sets in a single pooled experiment. One obviously needs to apply some multi-site corrections to these. But the objective here was to actually simply visualize all the pooled data that you had at your disposal and to really understand the patterns, what the data presented. So at Lonnie, we are developing an informatics framework which is going to integrate workflow design and optimization for different kind of data sets. So it's also going to involve different multi-site trials and extend neuroimaging atlases into different hierarchical spaces where one can actually load your data on the fly and visualize how it looks like. So we are also going to develop, or we are in the process of developing an interactive visual environment for the whole informatics framework, and so you can actually pull in your data, change different metrics on the fly, and then actually see how the data gets clustered. And so this will probably be available in the start of the next year for free download for all. So to summarize, our approach uses distribution functions of features for analysis. It doesn't require any pairwise registration. It's invariant to translation, rotation, and scaling. And since it uses distributions, it's fast, scalable, and it's applicable to large collections of brains, which we are interested in doing. In the future, this work can be extended into 3 different directions. So one is trying to do heritability and genetic studies for twins and other populations based on these distributions, and really trying to understand how various features such as thickness, volume, or area interplay genetically or even across a pedigree in a given population. The second is to actually use this representation for inferring networks, again structural networks purely based on thickness or volumes, and actually integrating both of these approaches into a large-scale informatics framework in the future. Thanks. That's all.
Time for questions. So let me start the question. So, did you have a chance to look at the reproducibility of your distribution functions, for example, for different collection of data from the admin, so, and also the selection of the admins? So.
No, so not yet, and we are in the process of doing that. So for ADMI, it's actually 384 sample, and we only selected 117 out of that for age matching purposes. And so you have wide age range, and I wanted to restrict the age range, first of all. That's the first thing.
Any other question? So if, let's thank the speaker again. Thanks.